the second chapter of class 12 biology, that is sexual reproduction in flowering plants. Hope you have gone through my first video where we have discussed about the male reproductive part, that is the stamen. Now in this second video we are discussing about the female reproductive part, that is called as the pistil. So we know that the female reproductive part in flower is known as either pistil or carpel, and collectively, that is the whole of this pistil, is called as gynoecium. Very simple, the three parts that the gynoecium has are the stigma, style, and the ovary. So one by one we will look at these specific parts. Stigma is the swollen tip of the pistil. As you see in this diagram, this tip, it's a beautiful photography, and at this tip you see this structure is the pistil. In fact, these small rounded structures which you see here are actually the pollen grains which have landed on it. So it acts like a landing platform for the pollen grains, and it is usually covered by certain sticky substances so that the pollen grains can easily adhere to it, that is, get attached to it. Next, coming to the style, the following part of the stigma, that is a long slender stalk which can be either hollow or a solid tube like structure, is called as the style. The basic function of the style is to keep the stigma attached to the ovary, and it also acts as a conveyor. Conveyor means something that carries. Carries what? Carries the pollen tube which is formed by these pollen grains from the stigma to the ovary. Now coming to the ovary, it is the last portion, that is the basal portion, which is enlarged. And the function is that it contains the ovule inside. It houses the ovule inside. And the ovule houses the egg inside. So in this diagram, you can see these are white structures inside are multiple ovules inside this single ovary. Now if we look at the cross section of an ovary, then we see the various internal parts of the ovary, such as the carpel, locule, ovule, funiculus, and placenta. Let us look at it one by one, the ovary cross section. Now, when multiple chambers of the ovary are present, if a flower has multiple ovary, if it is multicarpillary, then and the ovary, if they are all fused together, then we can see multiple chambers like this. In that case, the individual chambers are called as the carpel, and the outer wall is obviously the wall of the ovary, that is the ovarian wall. The empty space which we see within each carpel, these arrow parts, are called as the locule. Locule is also called as the ovarian cavity. Now in this locule you will find the development of the ovule. These white structures that you see here are ovules. Even here also these semi-transparent structures, oval structures, are various ovules, which is developing in this empty space, which is actually an empty chamber, that's called as the locule. When you have many locules present in a single ovary, we call it as multilocular ovary. Like here, this is bilocular ovary, this is trilocular ovary, and so on. You can also have a unilocular ovary where single locule is present. Further, if we look at the ovary cross section, then multiple locules, they join at one particular point here in the center, which is called as the ovary core. That is actually a solid structure that is present at the center. And from the core, the placenta appears. Placenta is the parenchymatous tissue from which the ovule develops. So this side, this area is the placenta. Like we see here a visual presentation, the central part is usually the core and surrounding part is usually the placenta. It is this placenta edge where the eggs or the ovules will develop. So placenta basically nourishes the egg. Further, in a cross section, we can see multiple ovules within a single locule. This is a single locule where multiple ovule may be present. And these individual ovules are attached to the placenta through a small stalk-like structure which is called as funiculus or funicle, or sometimes also called as funiculum. Now let us look at the types of carpel. Based upon how many carpels are present, if it is single carpel present in a flower, we call it as monocarpellary condition. For example, certain legumes are there. If multiple are present, we call it as multicarpellary condition. When it is multicarpellary, then two possibilities are there, whether the carpels are fused or carpels are free. If the carpels are fused, then we use the word syncarpus. And hibiscus, papaver, etc. are examples of syncarpus flower. If they are free, we use the word apo, that is free, apocarpus. For example, mycelia. In this ovule, we see the development of the future embryo. So the ovule is also called as megasporangium. Now understand the term megasporangium is the house of megaspore, and megaspore is what? The egg or the female gamete. So here actually the megaspore is not exactly female gamete, rather we can call it as a female gametophyte. We will look at the overall development, but first let us look at the structure of the ovule in general. Now, ovule has outer two layers of tissue. If you see these two layers, they are called as integuments. These two protective layers are called as inner integument and outer integument. Some species have only one integument and they are called as unitegmic. Usually in higher dicots we find unitegmic flowers, and some have usual two integuments, then you call it as a bitegmic. That is monocots and certain lower dicots have this single integument. Then inside that you have a mass of tissue, meristematic tissue, this dotted structure or shaded structure which you see, that is called as a nucellus. Now, nucellus is the tissue which will provide nutrition for the megaspore development. Now, we will come to this diagram in the center in our next discussion. Before that, this entire structure is called as the ovule. If we look at the integuments from both the sides, then we see that the integuments are developing from this area, which is called as a chalaza. From the chalaza, the two integuments appear on the opposite side. They do not join, leave a small gap. That gap or small opening is called as a micropyle. Now, this micropyle is the small opening through which the pollen tube will enter for the process of fertilization, that you will learn in the process of fertilization. Next, if we look at this outer part, we see this small stalk which is called as funiculus or funicle. This one attaches the entire ovule with the ovary. Okay, and from the ovary, this funicle develop and joins the ovule at this area which is called as the hilum. Hilum is therefore, we can say, the area of the ovule, the place of the ovule where the funicle attaches. And this funicle may go behind the ovule through the back of the ovule, forming a strong supportive structure which is known as raphe. Okay. Next, if we look at the different types of ovule, if we simply look at this diagram, you will understand. Basically, this ovule can develop in different design in terms of how it is arranged with the funicle. This is the one which we studied just now, which is called as a typical anatropous ovule. In almost 90% of the angiosperms, the ovule develop in this way. We'll start with this one called as orthotropous ovule, where the micropyle, this opening, chalaza, that is opposite, and the funicle, this funicle will be this side, they all lie in a straight line. Examples like Piper, Polygonum, and Cycas, you find such an arrangement. This one that is called as an anatropous ovule, here in 90% of the angiosperm families, we see that this entire ovule lies at 180 degree, where micropyle lies very close side by side with the funicle here. However, in this case, this is the funicle and the entire ovule is present at a 90 degree with the funicle. and this is called as hemianatropous arrangement. In this case, we see the ovule is slightly curved, almost at right angle to the funicle. Such cases we call it as campylotropous. In members of Leguminosae and Crucifer, we find a campylotropous ovule. This is amphitropous, where this is the funicle, almost look like anatropous, but actually the inner embryo or the ovule is taken a horseshoe shaped. It's curved to form a horseshoe shaped structure. Such an arrangement is called as amphitropous. Here you see the entire ovule has taken a 360-degree turn. From this is the funicle, and if it has finished in this much, we would have called it as anatropous. But it has further taken another 180-degree turn. That is 360-degree total turn. So this is fully coiled. In examples of Opuntia, etc., we find this arrangement of ovule. We call such an ovule as sarkinotropous ovule. Next, our development is embryo sac within the ovule, which is called as a female gametophyte, or we also call it as microspore development. Now, what happens is in the nucellar region, towards the micropylar region, usually certain cells they differentiate to form primary archisporial cell. Usually only one nucellar cell in the micropylar region differentiates. There can be sometimes more than one nucellar cell also. That single cell becomes primary archisporial cell which undergo mitosis to form primary parietal cell and primary sporogenous tissue, or initially sporogenous cell. Now, primary parietal cell do not divide further, unlike in case of anther where it develops into anther walls. Here the parietal cell is not dividing further. Here the sporogenous cell directly function as a megaspore mother cell, that is the diploid cell. Now that particular megaspore mother cell undergoes meiosis to form 4 megaspores, because one cell undergoes meiosis gives you 4 cells. So in this diagram you can see towards the micropylar end, this is a single cell which is called as a megaspore mother cell. This side you will find the other nucellar tissue. This one has started undergoing meiosis. After first meiotic round of division, you get 2 cells, and then again another round of meiotic division, you get 4 cells. With this one, we call it as megaspore dyad, and then it forms megaspore tetrad. In megaspore tetrad, 3 megaspore they degenerate; they do not develop further. This is very important to understand. And then only one megaspore will develop further. This phenomena is called as a monosporic development. Means the development takes place from only one megaspore. You see, this is a very important phenomena to understand. This is how naturally the plants ensure that only one embryo sac will develop, and therefore only one embryo will develop in future instead of 4 embryo. So only one megaspore develops into an embryo sac. Now let us understand what is an embryo sac actually. See, the embryo sac develops in this way. This is one cell, that one megaspore that we were talking about here. Now normally it is the 3 megaspore towards the micropylar end which degenerate, and 1 megaspore, this one will remain as towards the chalazal end for the further development. However, this one will undergo degeneration. This 3 will undergo degeneration towards micropylar end. Then that single cell which had one nucleus undergoes nuclear division, only nuclear division, to give you 2 nucleate stage. One more round of division gives you 4 nuclear stage, and another round of division give you 8 nucleate stage. These are all free nuclear divisions. So at the end of 3 rounds of free nuclear division, you will get a cell which is a single cell but 8 nucleate stage. In a single cell, there are 8 nuclei present. Then these individual nuclei are arranged towards micropylar end. The 3 nuclei will give you 2 synergids and 1 single egg cell. We will come to the function of synergids little later. This is important. The egg cell is formed, and flanking the egg cell there are two cells formed which are synergids. Let me remind you that in the previous slide when we discussed, this megaspore mother cell was diploid. So at the end of meiosis, the single megaspore was haploid. Therefore, here also all these nuclei are haploid nuclei. Therefore, here also the egg is haploid and the two synergids are also haploid cells. Synergid and egg together, they are called as an egg apparatus. Remember this term. Then, towards the chalazal end, the three nuclei will also develop into three different cells, which are called as antipodal cells. And in the center, two nuclei are left out with rest of the cytoplasm and the overall cell. This one further, if we look at, then it develops into this type of an overall structure which is called as an embryo sac. Towards chalazal end, three antipodals are formed. Towards micropylar end, two synergids are formed and one egg cell is formed. And the two nuclei which are left out, these two nuclei which are left out, along with the rest of the cell, form the central cell. Central cell therefore has two polar nuclei. Thus, at this stage, you will find 1, 2, 3, 4, 5, 6 cells and the overall central cell, that is 7-celled condition, where these three cells are carrying single haploid nuclei. The two synergids are carrying two single haploid nuclei. Egg has one haploid nucleus, but the central cell has two polar nuclei, both are haploid in nature. This overall structure is called as an embryo sac. Here the synergids have certain infoldings towards the micropylar end, which is called as a filiform apparatus. In brief, if I tell you the function, antipodals function is not well understood, but it is believed that they help in absorbing the nutrition. Synergids function is actually to guide the pollen tube during fertilization. You will understand that when we discuss about fertilization. Egg is obviously the main female gamete which is involved in the process of fertilization, and central cell with its two polar nuclei will develop into endosperm. Endosperm is the future nutritive tissue which will provide nutrition to the developing embryo. So this entire structure is called as an embryo sac. An important structure in the exam, you get question to explain the structure of embryo sac with diagram. Now overall process, if we observe, there are two processes we have discussed. One is megasporogenesis and embryo sac formation. So let us do a revision once again. Within the nucellus, one cell functions as a primary archisporial cell due to differentiation, which divides into two cells, parietal cell towards the outer side and sporogenous cell towards the inner side. Parietal cell does not develop further, it remains like that and slowly degenerates. The sporogenous cell acts as a megaspore mother cell and undergoes meiosis to form 4 haploid megaspores, out of which the 3 haploid megaspore degenerate and 1 megaspore remain. So here you see the degenerating megaspores and single functional megaspore. See, within the single functional megaspore, the nucleus has started undergoing free nuclear division, 2 nuclei, then 4 nuclei, then 8 nuclei. Due to 3 rounds of mitotic division of the nucleus only, you will get a condition of single cell 8 nucleate stage. From here, the nucleus are rearranged, 3 towards one end, 3 towards the other end, and 2 in the center. Then the 3 towards the chalazal end become the antipodals here. 2 towards the micropylar end become synergid and 1 become the egg cell. And the other 2 nuclei along with the overall cell become the central cell. These 2 nuclei of the central cell are known as polar nuclei. So this was all about the female reproductive structure and the development of the female gamete. So students, next class we will be discussing about the third part of the chapter, that is how gamete transfer takes place in the plants, that is pollination, and then how fertilization will happen in those plants. So that was all for this video. I hope you liked this video and understood this video, please don't forget to subscribe to my channel and like my videos, share it with your friends also, and keep viewing the rest of the videos for the other chapters. Thank you.